Glycolysis is the process in which glucose is converted into pyruvate. The process involves multiple enzymes and intermediate steps. These break the glucose to harvest energy, which is used to fuel reactions in the cell. In this video, characters in black attire represent carbon atoms, green represent phosphates, blue represent ADP, red represent ATP, and finally, the transition of the blue balloon represents an isomerization from glucose to fructose. Glycolysis begins with a 6-carbon sugar named glucose. This glucose is phosphorylated with the help of a kinase enzyme by ATP, which is reduced to ADP. This produces glucose 6-phosphate. The phosphorylated glucose is then isomerized into fructose 6-phosphate. Fructose 6-phosphate is then phosphorylated again by an ATP. This reaction yields fructose 1,6-diphosphate. At this point, 2 ATPs have been spent by the cell and no energy has yet been harvested. An aldolase is used to cleave the fructose 1,6-diphosphate into 2 3-carbon compounds, one of which is PGAL. The other is then isomerized into PGAL. NAD+, a B vitamin derivative, is reduced and later will produce approximately 3 ATP by way of the proton motive force of the electron transport chain. Each PGAL then has 1 inorganic phosphate added. These phosphates are then used to phosphorylate ADP into ATP. Each pyruvate produces 2 ATPs. Since there are 2 pyruvates, this means that 4 total ATPs are produced. In total, 2 ATP were spent to produce a net of 2 ATP and 2 NADH.